Science Journal for Kids and Teens presents How Can We Make Antimalarial Medicine Faster? Adapted from the original paper in the American Chemical Society and American Society of Pharmacognosy, published on June 17, 2021. Research conducted by Caitlin Varela, Hadi D. Arman, and Francis K. Yoshimoto from the University of Texas. See the full list of authors in the accompanying PDF. Read by Jacqueline Young. Abstract. For most people, getting a mosquito bite is annoying. For others, it could be life-threatening. Mosquitoes are carriers of many diseases, including malaria. According to the World Health Organization, in 2019, there were 229 million cases of malaria worldwide, and about 409,000 people died from this disease. This is why scientists are trying to make the antimalarial medicine Artemisinin more available. Artemisinin forms from dihydroartemisinic acid, also known as DHAA. In this study, we collected data to see how heat and light affect how fast artemisinin forms from DHAA. We also looked at how the amount and chemical composition of DHAA affect the formation of artemisinin. Our data support the need for an optimal amount of ultraviolet light to produce artemisinin faster. We also discovered that the presence of hydrogen isotopes, known as deuteriums, on DHAA slows down the process of forming artemisinin, but increasing the amount of DHAA can significantly increase artemisinin production. Introduction. Every 2 minutes, a child dies from malaria. People all over the world are working to stop this disease. Malaria is caused by a parasite spread by mosquitoes. To stop malaria, we need to control mosquito populations and produce medications that can kill the parasite that causes this disease. A natural medicine against malaria is artemisinin. It comes from a plant named Artemisia annua. This plant takes a chemical called dihydroartemisinic acid, DHAA, and turns it into artemisinin. Researchers are using chemistry to figure out how DHAA transforms into artemisinin so that we can make this antimalarial faster and in larger amounts. In this study, we wanted to find out which factors affect how fast artemisinin forms. Could heat, light, the amount of DHAA, or the chemical composition of DHAA hold the key? In this photo, we can see that sunlight helps Artemisia annua naturally make artemisinin, an antimalarial medicine. And this photo was taken by Francis K. Yoshimoto. Methods. Experiment 1. We place samples of DHAA under an infrared light, in a bath of hot oil, and under an ultraviolet light, also known as UV. The samples remain in their environments for 2.5 hours. We also put a sample of artemisinin under the UV lamp for 8 days to see how UV light can affect artemisinin. Then we measured the amount of artemisinin present in each sample using a nuclear magnetic resonance, NMR, spectrometer and a mass spectrometer. For our next 2 sets of experiments, we created a form of DHAA in the lab that contains 3 deuterium atoms, as seen in Figure 1. Deuterium is an isotope of hydrogen. The most common form of hydrogen has 1 proton and no neutrons. Deuterium has 1 proton and 1 neutron, so it is heavier than hydrogen. An isotope is an atom that has the same number of protons as other atoms of the element, but it has a different number of neutrons. Think about an apple, which may look the same as another apple on the outside, but has a different number of seeds on the inside. In Figure 1, we can see a comparison of DHAA with our DHAA with deuterium. Our DHAA has 3 deuterium atoms on the 15th carbon instead of the hydrogen. Here is DHAA without deuterium, and here is DHAA with deuterium, as seen in the highlighted portion of the figures. Experiment 2. We tested how fast DHAA with deuterium converted to artemisinin in a dark room, by a window, and under a UV lamp. We used a set of samples with 100 micrograms of DHAA and a set with only 10 micrograms of DHAA. Experiment 3. We mixed DHAA with and without deuteriums in a 1-to-1 ratio. Then we looked for artemisinin using a mass spectrometer. By mixing the two forms of DHAA, we can directly compare how fast DHAA turns into artemisinin when it has deuterium and when it doesn't. Results. Experiment 1. The UV light sample showed a larger amount of artemisinin compared to the infrared and hot bath samples. But when artemisinin was left under UV light for 8 days, the amount of artemisinin started to decrease. Instead, we found a chemical that had all the atoms of artemisinin, but in a different arrangement that does not kill the parasite that causes malaria. Experiment 2. In the first 24 hours, our DHAA with deuterium formed artemisinin 3.3 times faster in the light and 49 times faster under the UV light. However, at 120 and 312 hours, the rates of formation were about the same in the dark samples and the natural light samples. Under the UV light at 120 and 312 hours, the amount of artemisinin was 5.9 times and 50 times less compared to the dark. See Table 1 on the next page. There was less artemisinin because the chemical with a different arrangement was forming. We also learned that the amount of DHAA affects how much artemisinin forms. When we used 100 micrograms and 10 micrograms, we found that the vials with 100 micrograms made 39 times more artemisinin. Experiment 3. Artemisinin formation was faster for the DHAA without deuterium. Here you can see Table 1, results of the 100 microgram deuterium samples in the dark, natural light, and UV lamp over time. Right here, we can see the amount of hours that have passed, and over here, we can see artemisinin rate comparison, or how many times faster artemisinin is produced compared to the dark. We have 3 columns, one for dark, one for natural light, and one for UV light. What happens to the rate of artemisinin formation over time for the natural light and UV samples? Discussion. Our results show that we need large amounts of DHAA and an optimal amount of UV light to produce the greatest amount of artemisinin. That is, we need enough, but not too much, in order to get the maximum amount of artemisinin as quickly as possible. You can probably think of some things in your life that work like that. For example, you need to eat enough food at lunch, but not too much in order to have energy for your afternoon classes, but not fall asleep. Artemisinin forms faster in UV light than natural light, while heat doesn't help at all. But if the DHAA is under UV light for too long, it forms a new product that doesn't have antimalarial properties. Also, 10 times more DHAA created 39 times more artemisinin. We also found out that artemisinin formation was faster for the DHAA without deuterium. You might think that that means we wasted our time making the isotope with deuterium, but actually, it's always useful to find out what we shouldn't bother doing in the future. Conclusion. Scientists are working hard to make treatments for malaria available at a low cost. You can help through donations to organizations such as Nothing But Nets. They send mosquito nets to countries in need. Make sure to protect yourself from mosquitoes too. Wear long sleeves and pants or use insect repellent when you are in areas with mosquitoes. Also, make sure doors and windows close properly so they can't get into your home. Even if you don't have to worry about malaria, there are other diseases mosquitoes can carry. Thank you for listening to this recording. Visit our website, sciencejournalforkids.org, for more free science teaching resources.